Okay.
Thank you very much, Alfonso, for agreeing to give a Build a Cell seminar, and you can get started.
Okay. Thank you very much, Kate, and thank you all the participants and online participants for this opportunity to present my work. I mean, I hope that it's going to be of interest to all of you. And so I'm going to talk about an adaptation to complex decisions, how bacteria could adapt and learn things that are complicated. Okay, so usually, this complex decision making, okay, requires careful design, but this is too complicated. So, and we don't know living systems that much. So here the goal is to do it through an iterative improvement. So the system, little by little, is going to adapt, and then it's an iterative process, and we, in this way, it's going to just learn. Pretty much like today in complex neural networks, how they learn. And normally for to have living systems, in particular bacteria, we are here concerned with bacteria, and to have a given behavior and decision making. And normally you use gene circuits, and in gene circuits, most of the information normally is encoded in the DNA. So you design these gene circuits, and then they will behave forever like that. So it will behave maybe oscillating, maybe doing some pattern recognition. But if you want to change that behavior normally, you have to design it again, okay? And normally, if the system you want to behave, it has to be, it's a complex system, then you need a lot of truth tables, and which means a lot of so you need a complex truth table and a lot of logic gates to implement it, and therefore a lot of transition factors. That is one way to implement logic gates in biology. And but this is very complicated. You can use somehow, I mean, switches and other types of dynamical systems where your behavior of a gene circuit is changed on running time. So it's just when the cell is already, you have cloned it, and then it's there, and then it's confronted to the environment, and then you can change that behavior. But then it's a discrete behavior, and it's very difficult that you can use that to for complex decision making. So how are we going to do this decision making? We are going to do this in engineer an multicellular sensor system that's going to be able to make E. coli to adapt to chemical patterns. So we are going to have a population of E. coli cells, and then they will just change their behavior according to a chemical pattern. And so in this particular, we are going to do is they are going to express a red fluorescent protein, and then the cells are going to be engineered to maintain this red fluorescence through many generations, okay? At least and as many generations as we need for the an experiment, okay? And then they will use chemical patterns to adjust permanently the expression. This means that if the cells to a given chemical pattern, they light to a given fluorescence, then they can be trained to express less red fluorescence to the same pattern, but not to other patterns, only to that pattern, okay? So that's a very key and important, because if we are able to do that, then we open a lot of possibilities in programming decision making. Okay, so then, and here, and I have, and normally, in gene circuits, you would use for when you design something that is very complex, you use directed evolution. That's a phenomenological way where you can adapt a gene circuit not knowing what DNA encoding is the most appropriate. Okay, and that is also the framework we are going to use. But we don't call it an evolution somehow, in the sense not to confuse people with Darwinian evolution. Okay, so we call it adaptation. So, but you can call it evolution. I don't know. So Darwinian evolution goes normally, there is a more discontinuous movement in phenotype space, because you get mutations, and then sometimes one single mutation, you can have a drastic effect in phenotype. Okay. And it's slow, so already the probability of mutation is low. You can increase that, but then you have challenges of with the organism, I mean, killing the organism. But also the problem is that the mutations may not go where you want to go to be faster. So, and then it's very difficult to cover the whole genetic space, because it's too big. So then, it's too daunting, the problem. And so then our approach is going to be to create what we call a phenotype at the bacterial population level. And so let me explain. We are going to create use allele frequencies to work as a genotype. So this means that the amount of the DNA is going to be the genotype. So we are going to use the concept of plasmid heteroplasmy. That this is when you have some bacteria that have a plasmid, and then they get some mutation, and then this mutation is neutral. So then the plasmid remains there, the mutant, even across the replications. So the frequency of that plasmid has been shown, and this was the firstly realized by David Liu at Harvard a few years ago. He published that in a paper, and that it was more, he used it in base editors, but he introduced the concept of what he called analog memory. And then even when you have when the cell of course divides, you have equipartition errors. This means that not all the daughter cells have the same amount of plasmid than the mother. But this is a good thing for us, because these are it produces some diversity, which is we need for in order to search the space of decision making. So we need some randomness in order to explore, to better explore the space. Okay. Then we are going to use to modulate the frequencies of these alleles in sublethal antibiotics. Okay. So then this is like in fact, this is a selection for not killing, but by making some cells to grow less well than other cells. Okay. And then at the end, you have the proportion of some cells that are enhanced and increased. And in this way, I mean, you modulate this genotype. Okay, we'll see all this later. Okay. So then, as I said, we are going to do this an iterative improvement method, and where the cells are exposed to chemical patterns. And in this way, what we are going to do is to use the sublethal antibiotics to punish incorrect decisions until so the cells, what they are going to do is they are going to each time behave in a different way, but avoiding the last punishment. So the last behavior that was punished. So, and we are going to demonstrate that through a simple game. That the advantage of this game is tic-tac-toe is that most people know this game and the rules of the game. So you have to have three, and I mean, the player that has three on a line, I mean, three positions on a line, then wins. And it's a game that it's I mean, from already the Egyptians, maybe 1000 BC, it's there. So, and then it has been used as a benchmark in the first computers like ENIAC after Second World War. And so in our case, and this is a little, we are not programming the cells and to play tic-tac-toe. No, we are teaching the cells to learn to program themselves to play tic-tac-toe. Okay, so we don't know what, I mean, we could know if we do simulations and then do a lot of studies in order to know what are exactly the genotype that we would need in order the cells to play expert for tic-tac-toe. This is too difficult. So what we do is we use this punishment procedure that it's a type of reinforcement learning that I'm sometimes scared, because then the reviewers are always very picky on when you use the terminology that is already very, already solidified in some disciplines, like in computer science. So then I will have to be careful. But okay, I'll use it, and then I excuse myself if it's not what other people think that is what it should be in reinforcement learning. Okay, so then now let's go a little bit more in detail. So that I more or less I have summarized what I'm going to talk. So somehow, so now I'm just going to tell you exactly how I do it. Okay, so first thing we are going to do is we are going to engineer the cells having two plasmids. The two plasmids, and we are going to call P1 and P2. The P1 has a red fluorescent protein and cherry that is has to be made to be same size than this green fluorescent protein, enhanced GFP in the P2 plasmid. So the idea is that these two plasmids have exactly the same size and exactly when the same replication. So this has something that normally nobody would do, that is to have the same resistance. Ampicillin in this case, I'm not showing that here for simplicity, and the same replicon, a coli 1. Okay, if you have this, normally you have what is the same compatibility class, therefore they will be incompatible and one plasmid will just get kicked out of the cell. Okay, but here is not the case, because the plasmids are indistinguishable for the replication machinery and for the genetic load. Okay, so this produces that they can coexist. Okay, and that's very important because that's the basis of this type of memory. Okay, because this is a multi-copy plasmid, and then you will have many copies of P1 and many copies of P2, and those copies will remain. So, and in these promoters, they will have the same promoter. They could have a different, but then these are other types of applications. But for what I'm going to tell you today, it's the same promoter. And we take a promoter from the marionette cassette from Chris Boyd at MIT that he engineered a few years ago, a strain that can express 12 transcription factors. We have chosen 9 of them, and these are listed here: salicylic acid, ATC, etc. All these. I put this short letter just not to crowd the slide. And then this makes this system to be a sensor, because these are the transcription is activated when only when you have the cognate chemical. Okay, so if I put here the PSAL promoter, then only when I have salicylic acid, it will get transcribed. And then I use a ribozyme, just an insulator, a typical insulator in 5' UTR, so they can be more standardized all the translation. Then I put in a cassette of antibiotics. So this is polycistronic. So this antibiotic cassette, this is in polycistronic to the fluorescent protein, so that you have a second RBS. And then in order, here you have a kanamycin resistant gene in P2 and in P1 a chloramphenicol resistant gene. But for the sake of having exactly the same size of the plasmid, and then we have just used this fusion. This was already proposed by David Liu, so then we just copy that design. Okay, so that we thought it, but in our case, it's not really necessary in the sense that in this promoter is inducible. So normally, the cells, this promoter is not induced, therefore, and they have little leakage. So this plasmid, because the cargo is not transcribed, then it's not a big problem that here, meanwhile, the size is the same, then it's not so big issue for genetic load, because there will be no genetic load because the plasmid is not transcribed. This promoter is not transcribed, okay? Contrary to this memory of original memory by the Liu. And okay, so then we tested the stability in liquid medium, and then here is an example, but I mean, we have done for all the promoters, and also in solid medium. In solid medium, you have bigger error bars as expected, and but it's much easier to show things because it's more visual, and then you can do more in throughput because you have the plate, and then you have the tic-tac-toe board, and then they have the cell, so it's much easier to do like that. So then we have used that, but we have also done it in liquid medium, and so we are going to define what we call weight, that is the fraction of the P1 plasmid. So remember that we said that the P1 plasmid is the one that encodes the M-cherry protein and the chloramphenicol resistance, and the P2 is the one that encodes the EGFP and the kanamycin resistance. So the weight is going to be this number of copies of the red, this X, divided by the total number of copies, which is the plasmid copy number, and which is constant across cell replications. Okay, and okay, so then what here is interesting to show is that this the denominator is constant, but also the numerator is constant, and then this weight is shows that this constancy. Okay. Okay, so then another thing it's important is that is the population average of the weight that is constant. Okay, so the cells still, when they divide, then they will it will, as I said, there is an equipartition, and then so there is at the single cell level, it's not going to be the case. Okay. Then here we tested, I mean, this here, I don't know what is the best way to, I mean, to plot this in a way that is easy to check. So this is in solid medium, and then we have 3 replicas for every promoter in a different color, and then we just the measurements. When we do the measurements, what we do is we have the bacteria in solid medium, so then we do a replica plating, and then in the replica plating we induce, and then we wait for the cells to get fluorescent, and then we measure the fluorescence in red and green. And then when we do that, I think I have a slide. No, I think I have it later. We measure in red and green, and then we compute the ratio, because we cannot normalize by OD, because in a solid medium it's very difficult. So we use 2 fluorescents, and then we compute the ratio between them, and then from that we can get the weight. Okay, so and we also did it a single cell. So there's a single cell, and then is in liquid culture. Then we go through a flow cytometer, and then we just analyze the different plasmids we have. I mean, the different bacteria, and that have these 2 plasmids inside, and then you have in the P-cell in a system, that this means that the bacteria that has a P1 plasmid with the P-cell promoter, and P2 plasmid also with the P-cell promoter, that one in red and green. And then when we use, in this case, we have used kanamycin and the inducer. So you have to put the inducer as well, because otherwise the resistance in a protein is not expressed. Okay, that's important, and that's usually somehow people are not used to that concept of having an antibiotic, a resistance that is depending on and is conditional. Depends on the inducer. Okay, and this is it produces is the key of producing some adaptation. So this means, for instance, that here, if you put in a kanamycin, then you are selecting the cells that have a lower weight, because lower weight means more of the Y or less of the DX, and it means more of the kanamycin resistance. So then those cells grow better, and then you can see here in repeated flow cytometry experiments of a different after incubating with kanamycin, sublethal kanamycin. So I mean 10 times less than what you used to, in plating. And then you can see that the average value of the weight decreases. Okay, in all cases. Okay, so that this we can use then the same thing chloramphenicol, then increases. I'm not showing that, but and then for the same reason, because the with chloramphenicol, then you are the cells that have more of X will increase their number. Okay, so this is like some type of Lamarckian behavior, that is, the more chloramphenicol resistant you are, the more you can be resistant afterwards. So then you put the cells chloramphenicol, and then they get more chloramphenicol resistant. You put more chloramphenicol and more chloramphenicol resistant. Okay, so then, I mean, without getting any mutation. Okay, so but this is reversible. So then you can just go with kanamycin, and then they go back, okay, to the sensitivity. And remember, this is at the population level in averages. Okay, so because this gene expression now depends on the history of doing kanamycin, I mean antibiotic selection, I mean this sublethal selection, then this gene expression, we say that is dependent on its own history. Okay, so then it's also important to know that, for instance, it's very important if you leave the cells may, I mean, if they have some background expression of they can change potentially, I mean, their weight. So it's very important to be careful when manipulating. But that also happens very often with gene circuits. Okay, so that where the state depends on the concentration of transcription factors, and then and you have to be careful when you manipulate. So we have named that memregulon, just inspired by memory and regulon, like memristors, where you have a component, electric component, that it's very much used also for neuromorphic computing to simulate neural networks in electronic devices. Okay, so then we name it like that. Okay, and then okay, so then here it's a little bit a summary of the specification for this memregulon. That is, you have two plasmids that have the same original replication. So here the original replication is coli 1. Okay, it's not the same, and same antibiotic resistance. So here is white. Okay, the original replication, and here in this green thing, or bluish and green, you have the ampicillin resistance. So, and the inducer I, because we have 9 possible inducers, because we did a battery of these cells, activates the output gene expression. So it activates the red and the antibiotic resistance. In this case, is, remember, chloramphenicol, and then the green here, and you have here kanamycin resistance. And gene expression is proportional to the number of DNA copies. Okay, so this is typical central dogma thing that the more DNA, the more RNA, the more protein, and then and here we are regulating gene expression, but at the DNA level before going to the transcriptional level. Okay, and then in here this regulation, contrary to transcription regulation, in this regulation is permanent. So you change it and then it doesn't go back. Normally with transcription regulation, you have a transcription factor stops, then you go back to the where you went. And in fact, it's more difficult. So normally there are other types of memories. I mean, normally memories and you can have in the number of a molecule. For instance, a protein. You can have some ion channel in a synapse, and then you can have more of those ion channels. But if the cell doesn't replicate, then this number can remain fairly constant, and that's fine. But in bacteria, cells replicate all the time, so you cannot have a memory on the number of proteins because that will just get diluted away by cell doubling. So then you will have to have a replicating molecule. So that's why we chose in a DNA, having a plasmid as the source of memory is very convenient for systems that are replicating, okay, all the time. Okay, so then if you get to have a cell that doesn't replicate, and then we could think about other other types of memory systems. Okay, so and here we did, this is in a liquid medium, and then we did the characterization of the learning of the system, and we compare with qPCR. So on the right is on the qPCR, so we just measure the amount of with primers for P1 and for P2, and then we see the proportion of the P1 over the total. And then, but we can also, on the left, we measure the promoter activation, only on the red versus the background. So this means when it's not induced. And then here, for instance, I'm just amplifying one case. So each of the case, you have several cycles. So here on the right is only one cycle, but we did 5 cycles of learning. And then you see that the cells in all the replicas, they change this the weight. Okay, and it agrees pretty well doing full fluorescence done with qPCR or doing some calibrations, because you can just choose a right amount of the plasmids, I mean, or population of cells, and then you can benchmark that. It's very easy to do that and then to calibrate the system. Then we also did some combinatorial promoters with these were not existing in the literature, so we engineered them by just putting operator sites in the promoters on the right of the -10 sequence, and just to create a combinatorial. That's not easy, so we only succeeded in 4 cases. Okay, and we succeeded more, but only in 4 cases in P1 and P2 plasmids. Okay, but I'm not going to use here in this talk. Okay, so then another, we have then, as I said, we have a system of 2 plasmids in one cell with only 1 promoter that says one thing, that's one of the 9 chemicals. But then what we want to do is to have sense the capability to sense a pattern, but a pattern up to 9 chemicals. So then we are going to use a consortium of cells. And then here we use this paradigm of multicellular computing, or distributed computing, that where you distribute a gene circuit across a population. So each cell does a different task, and then they all you combine the output of the cells. Okay, in this case, because it's fluorescence, it's very easy to combine, because then you just in fact you see the fluorescence of the population. So this is a concept that was put through by Ricard Soler in Barcelona and by Chris Voigt at MIT. Okay, and then so it's very useful in order to create complex systems, complex gene circuits. So, and then here what we have is that we can have this type of OR system. I mean, I put it like that, where you have all these inducers, and then you have as output red fluorescence and green fluorescence. But what we do, and then we here we do it in a when we analyze the data, is we apply an equation that this is nonlinear. So you see, this is exactly how it looks, this function, to create this what we call F weight, which corresponds exactly to the weight in a single, in the case of a single population. As I showed you before we just characterize with fluorescence the weight, and we characterize with qPCR, and then we got a very nice agreement. But also we characterize with sequencing and etc. So you can do it. We did in fact with Sanger sequencing, which is very low cost and easy. Okay, so yes, with the chromatograms, you just can play with that, and then you can infer the ratios in this type of degenerate chromatograms. Okay, so and here, so we have this system. So then we can have what I call multiplex adaptation. So in this multiplex adaptation, it's I mean, the idea is that you have several inducers at the same time, because you have several cells. Then it's not a problem. Each cell knows that it will get only activated by its cognate inducer. And then if you have kanamycin at this time, then what will happen is that they will just decrease their weight. So the SAL1 will just decrease, but then the CHO and the NAR. Okay, so these are, and the others will not. Okay, and that's remarkable that the others will not, despite of this sublethal antibiotics. So there's just the decreasing in growth, but then the decreasing growth equally. Okay, so then this is the function. In fact, let's see, this is the function that I have in this nonlinear function I said, that is just a function. You can derive that easily and mathematically to convert from ratios of green and red fluorescence to the amount of P1 plasmids and P2 plasmids. So this takes into account about autofluorescence, etc. And then this what we do, as I said before, to measure, we do replica plating, so we don't change what we are measuring. So we don't, I mean, of course, when you do a replica, then you change a little bit the plate. So in fact, we let it grow when we have this parental and we do a replica, either to compute the fluorescence or to compute the DNA quantification, just in a qPCR or sequencing, and then the parental also will allow to grow it for a few hours. And when we do adaptation, then when we do it in liquid medium, just put an antibiotic and the inducers. But if you do it in a solid medium, then we do again a replica plating. The reason is that we need to stop to have a dilution. So then, and we need also to have, when you do the replica plating, the cells start growing, and then they're more sensitive to the antibiotic, and then you have this sublethal antibiotic taking place. Okay, so we need growth in order to do our sublethal antibiotic thing. Okay, so here in the parental you don't have, it's already stationary, so that's why, I mean, you need to do a replica. But then we do another replica to stop. So here you have inducers, but when we do the second replica, we don't have inducers. So then after this replica platings, then you have a new parental. Okay, so then in measurements, we don't change the parental. It's just we make copies of the parental and then we throw away the copies. But in the adaptation with antibiotics, then we change the parental. Okay, so that's the important concept to remember. Okay, so then we do two replicas. So this we throw, then, but this we keep, and this we throw. Okay. So then we can just measure this inducing with the non-cognate inducer, and then etc. And we see that in possible crosstalks, and then we can and the crosstalks is around less than 10%. In fact, yeah, less than 10%. And then this is for the weight. Here is all these measurements are the weight, and then are multiplied by 100 just to make it 13. So in fact, when you see 13 here is 0.13. So this means that the weight has increased a value of 0.13. Okay, and then you see that for the red one, this is for chloramphenicol. Chloramphenicol increases the weight, because remember that chloramphenicol was in the P1 plasmid, and the kanamycin decreases the weight because it was in the other plasmid. So, and the weight is a fraction of the first plasmid, so then it decreases. And so then here we can just do a few things that are just to warm up on this type of computing. This is like learning a digital alphabet. Okay, so then there has been work by, for instance, Chris Boyd on creating a gene circuit with a lot of logic gates, and I think it was 70 kilobases and DNA footprint, where to create a digital display. Okay, so then one way to create this is just making not an encoding, I mean, 70 kilobases of gene synthesis in the cells, okay? And then you have to design that computationally, and then, I mean, with all these and cello and other softwares. But no, and one possibility is to have dumb cells, and then the cells to learn. Okay, so then how would this work? You just write the pattern. Let's say you have an S as pattern. So then in these cells that you have cultures, here you have 7 cultures. These are placed like in a digital display for simplicity. And then when you put in this, and you put the cells, then you put in arabinose and chloramphenicol on only this cell, this cell, this cell, this cell, and this cell, making the shape of an S. Then when you do this, there, in fact, what you do is a replica plating when you do the change of parental, and then what happens is that the weight of only for the arabinose membregulon will increase, but not for the others. Okay, so what happens when you do that is that you do this, then now you have another day, and then you just put to culture, and then you have the cells. But of course, here these cells, they have a higher weight for arabinose than these cells. So when you induce, then you get back the S. Okay. Now, you can change the cells to learn the same cells. You can learn all the letters. I mean, here you just learn CSIC because this is my employer. So then, to please, I mean, the big bosses, then it's always good to put the acronym of who pays you, and then these cells are induced with the cognate. Okay, so here and here is the same thing. Okay, but then the cells also can relearn the things, because then suddenly you say, okay, this S, now I want to learn another thing, and then you can learn to be an L. Then again, there are an L. Okay, and then if you put arabinose, it's still an S, but if you put an sal, then it gets an L. Because as you can imagine, when you put the sal, then the cells here, they will have sal, the weight increase, but they were already increased for the C, so for arabinose. So then you have 2 weights that are increased. Okay, so you can realize here that by having several weights in each population, then you can remember different chemicals, and then you light according to that. Okay, so this is a very simple thing. Okay, but then you can go and exploit that much further. Okay, so then here is we just relearn that to be an LAI, standing for Living Artificial Intelligence. Okay. Okay. So, but we can go much further. Okay. So, in order to go much further, is that we need to realize that we have the weight is almost a continuous function. I mean, it's a continuous values and an analog memory, more or less. I mean, it's not exactly, but and then so this is powerful because it can capture much more information. It's not a Boolean that you have a lot of the weight, so it's much higher than the other guy, and that's it. That's it. For that you can get an alphabet, yeah, you can do that, but but you cannot capture all the information to master some complex decision table. Okay, and that's what we want. So then, and for instance, the tic-tac-toe table. Okay, so then for that we are going to arrange this mapping of position of the board to the inducers. Okay, so these are going to be the positions of the opponent. So we are going to have the cultures of bacteria. These are co-cultures. So these are 9 membregulons for each of the 9 inducers, and these inducers, in fact. And then we are only to record the state of the opponent. So this means that the bacteria doesn't know their own state, okay? Only the opponent. But that's this simple algorithm is already good enough in order to achieve a mastery. Okay. Then, and what we are going to do is, we are growing all the cells and with the appropriate inducers. The inducers, these are corresponding to the opponents in position. And notice that here I forgot to put a culture in the center because I did it on purpose. The idea is that bacteria is going to play against an opponent, and the opponent is going to play first and is going to play in the center. So this is the most difficult situation for a player, because you play second, and so your only chances are of drawing against an expert. Is you can only hope to draw in a maximum, and then your percentage of success is very low. Okay, so that would not be the case if you were first player. So we wanted to have more margin to learn, and so then the bacteria starting from tabula rasa ignorance, so it's completely plain random. Okay, so then we grow the bacteria in the appropriate inducers, and then we compute. We induce them, so then we allow for a few hours for incubation, and then we compute the fluorescence in all of them, and we compute this F value, which remember is a nonlinear function that and it's aimed to infer the weight of the system. Okay, and then just we take the one with the highest weight. In this case, is this in a 4th position, let's say. If you is 1, 2, 3 like that horizontally, and then that would be the decision of the bacteria. So that's where the bacteria would move. Okay, so then we encode decision making, okay, by choosing the highest fluorescence among cultures. We have 8 replicas of the culture. At the beginning, it's the same culture, 8 replicas, but then they will just go and adapt each one independently, so therefore they are not replicas anymore, they are different. Okay, so in total, just just to remind you, just to also focus you, is you have 9 weights in this culture, 9 weights in this culture, therefore you have 72 weights in total. Okay, okay, so then in fact, just let me say, this is equivalent to a 1-layer neural network with 72 weights. Okay, okay, so, and then now we are going to do this iterative process where you have the bacteria that you induce with the opponents and inducers. So if the opponent is in these 3 positions, occupying these 3, I'm not showing the bacterial position. Of course, if your opponent has 3, the bacterial will have an 2, and then because the X starts first, so then it starts here, so then we'll have still 2, but I'm not showing. And then you just induce the positions here, the cultures, and the move is the highest in F, okay, the highest fluorescence, let's say, F weight. And then this is what is the position. Then the environment is the changes, because then now the environment is the opponent, human or machine or whatever, and then the opponent as a consequence of the bacteria playing a position, then it moves to a given position of the opponent's choice. So now the chemical pattern changes. So you have the previous pattern, chemical pattern, plus another inducer added. So it grows. Okay. So this is an example here. You have in this inducer here at the beginning, because the opponent plays 5, then automatically, and you put the inducer 5, that is this green color. Then you put it in all cultures, okay, except 5, because of course, so all the 8 cultures. And then here, it happened that it was the 4th culture that has highest fluorescence. So I'm showing the fluorescence, the red fluorescence, the F, and for a given inducer, after inducing with a given inducer by this tube, okay, the length of the tube here. So this is the longest. This is shortest. Okay. Then it's second round. So here it's position 4, and then so it's here. Then human opponent or whatever plays first, position 1 here, and then bacteria. Then what it does is you now induce with 2. Okay, so position 5 and position this color, that is color corresponds to the 1. Okay, so 5 and 1. Why? Because it's 5 and 1. And then you induce all the cultures. And now the cultures will light, and the fluorescence is the addition of these tubes. So this is the lighting due to the cells having a weight for 5. And then this is due to the weight for 1. But then you see that, for instance, in this case, this is the longest sum. Okay, so then it chooses this position 8, which is this one, 8 here. But then wrong thing, because the human on the opponent chooses here and then wins. Then when it wins, we do reinforcement learning. What we do is we to the all decisions. So decisions were here. This is a decision where you put all this growth, and here. So there are 2 decisions. Then we repeat the decision, but with the antibiotic. So this is the this antibiotic acts, you may think, as a type of dopamine or something like that, or I mean, something in a neurotransmitter. I mean, something that it helps the system to learn. So then in the same environment than before. So then what happens is that now when you have asterisks, you only put in the cells that have in the decision. So you put in this cell number 8, you don't put any antibiotic in the others. So of course, then that will change. And then you put it in the 4. But of course, then you will have changes in 4 and in 8, in 4 and 8. So when you induce with 5, of course, the one induced with here, you will have much lower. Okay, and also this much lower. In fact, it's so much lower that now it chooses the first. Okay, and then here you see that here is different because you have another inducer, so you cannot compare with this. Okay. And then what we see here is that the game continues in a different way. So it goes through a different decision, okay? And that's the key thing, okay? And so here, I'm not saying that we have a very nice way of optimizing, behavior by a convergence towards an optimal behavior, like when you do back propagation, then you know how it should go, and then you just enhance, you minimize the error, and then you just go in one direction. No, here we just we punish that behavior, so the system will avoid that behavior. So then it's going to sample all the possible behavior. So in a finite decision tree like this one, in fact, here it has very few possibilities, then it can just get to converge to an optimal state, which is the only branch that is for mastering. So we are going to see this now. Okay, so then here we start the system. So and then we opponents placed in the center. So we have all the cells here, and then you just the highest F, it was in this cell. So then this is this choice. Then we just apply an opponent's place and the bacteria place. And then again, we put two inducers. Remember, or in fact, here we do 2 rounds. So 3 inducers, because there are 1, 2, 3 positions. And then when it loses, then we do canamicin. And then what we do is we compute the expertise. So here is random. So we call expertise is just we play, and we do a simulation to see with the current weights of the system, what would be the amount of games that it would win if it plays with a random player 100,000 times. Okay, so then you get really a good accurate value of the expertise. Okay, so then we just do all these games, and then you do all this has been chosen such that so the expert player, I mean the trainer, we call it, I mean it's always always wins, and it makes the bacteria to learn appropriately. So then to minimize the number of learning. So otherwise it can be like 50, minimum 50 or something like that if you don't optimize. And then here you see sometimes, for instance, that expertise decreases. By no means this method will always increase expertise. Okay, so it's true that you don't make the same mistake as before, but you may screw up some weights for the others. So it's not obvious. Okay, so then after that, you get expert player. Okay, so then we can just so this means that it wins all. So then we did experimentally playing different branches. Okay, just to show that it's it wins. Not only verifying computationally that it's expert, but also experimentally with playing all possibilities. Not all possibilities, selection. And we did also negative control, just doing the incorrect learning. This means just using chloramphenicol instead of canamicin. And then we also did some rational weight, but I'm not going to enter. Okay, so we also computed the stability in time. So the system, when they have learned to master tic-tac-toe, they remain that. How long they remain? So we just tested for 60 hours and several freezing in minus 80, and then they could just maintain that. And then I'm not just going to enter in this. This is more or less a summary of what we did. Okay. We also did some pattern recognition, but this is more for the future, so I'm not going to enter into that. So I'm just going to go to the conclusions. I'm already almost an hour talking. So, and I have shown you a library of gene circuits that are very minimal. It's just what it would be like a yes gate, just a sensor system, but using intracellular plasmid mixtures that can autonomously adapt to target behavior. So this means autonomously means that you don't need to know how, I mean, what weight they have to have. Okay, so that's it is adaptative. Okay, this is I make this remark because there has been in last year a publication in Nature Communications of a neural network where very nice, I mean it's a nice read, but here what they did is they design computationally the weights, and then you create the given network, and then it behaves as is designed. Okay, other networks have been done. I mean, with DNA computing, it was a few years ago, where for handwriting recognition, it was also designed and manually, rationally. Okay, so here the thing is that this is useless. If you want this to have an application, you don't need to have an analysis and then simulation facility in order to know what should be the amount of weight you should put in your population. I mean, modify or set in order to have a given behavior, because this is the normal way of behaving with gene circuits, where these are a different type of gene circuits that adapt to new environments and to changing environments by adjusting the gene expression by learning on the way. So this dynamically, they learn on the way, like a brain. So you get this type of memory, and they change the behavior. So you have, so we think like our system as a building blocks, then you can reuse it for different behaviors. So in fact, our analogy for neural networks is not, I try not to use this word in order not to say that this is interesting because it is a new possible implementation of a neural network. No, this is the only point of the analogy with neural networks is the capability of extension, of how to inspire people on how you can build from that much more complicated things. For instance, multi-layer systems. Of course, that requires cell-cell communication and all that. So, but already you know what it requires. So then, and that's why, and I mentioned this in analogy. Okay, so let me just thank you all of you for you are listening. So when you are talking online, I hope that I still have internet connection. I'm not being talking alone. Okay, so I have postdoc positions, and I have all types of positions. So to work on this, but okay, if you want, please send me an email, alfonso.jaramillo@csic.es, and then. But other emails I put in my publications also, they all work. Okay, let me thank my people, and now we are starting from scratch in Valencia, so we have a few people, but we are going to grow very much. Thank you very much.
Thank you so much, Alfonso. There are some questions in chat. Can you see chat?
Yeah, okay, I see. Okay, let's see.
If you can read the question out loud before answering, that's going to go on recording.
Okay. So, a question by Brian. And the memregulum concept is fascinating. Is this system limited to antibiotic selection, or could you engineer a similar thing with other selections, like, for example, biosynthesis pathway for a crucial nutrient? Thank you very much for the question. Very nice question. So I'm very glad that to have these thoughtful questions, and yes, it can be used with other systems. For instance, if you have a biosynthesis pathway, okay, for a crucial nutrient, then the crucial nutrient, you need to have a sensor for that. If it is crucial, surely you can make a sensor for that. Okay, so and then this sensor, and you need to, for learning, you need to change the ratio of P1 and P2 plasmids. So before it was with genetic selection, but you can use, for instance, and that's something that we have we have done in the lab. I haven't published that. Is with this CRISPR-I. So you can downregulate the amount of plasmid, okay, by using CRISPR. Yeah. Okay, so you can use CRISPR-I or you can use CRISPR. So it's a choice. Okay, and you could also use other systems. I mean, okay, you can use transition factors, etc. Okay, so that that also we have used. So that's a way in order to have other things, antibiotic selection. And okay, another question by Bernard: does the memregulum system account for metabolic load? Higher expression and higher plasmid replication puts a strain on cells that might affect fitness, right? And or is it not significant enough to affect the outcome? Okay, very nice question as well. And yeah, so the thing is, we have made a lot of care in order to avoid metabolic load. So in fact, it's true that higher plasmid replication puts strains on cells, but the thing is that the same high replication is for the same two plasmids. Okay, so the plasmids normally during the memory period they are off. So they are, I mean, when they are not activated. So the memory is maintained because the metabolic load by both plasmids is very similar because there is no transcription from the plasmid. Okay, you have only the original replication and the ampicillin, but this is exactly the same thing. Okay, so then you don't have. Okay, so that's this has been done in purpose. Okay, so it's true that when you have antibiotics, then for instance, chlorphenicol also affects the copy number, but then this is during learning, and then this has been already fine-tuned during the learning process, so during the calibration. So okay. And another question by Mohammed, and a similar topic to the previous question. Have you, other people, seen how many plasmids you can put inside E. coli before you see clear burden effects? Assume you have an average strength promoter, but I'm curious to see how promoter strength affects it. Okay, and yeah, okay, so I think that yes, there are some in old papers I've seen. I've seen it, and then I don't remember where I saw it, that they had put even 11 plasmids on the same from different 11 different compatibility classes. Okay, so then so you can have, for instance, David Liu normally in his space system uses 4 plasmids. We also use 4 plasmids in our phage replication. So you can use, I mean, coli 1, P15A, PSC 101, and you can call A, you and PBR, PBR, and I forgot the one that from Bordetella is a broad range, RSF 1010. There are many origins that are compatible among each other, but let me just ask and answer a little bit more. The question is that the burden, in fact, would be the total amount of DNA copies and expression. This is true. So, but the plasmids, they have a copy number control. So coli 1, they have only an RNA that express like P15A. So then the burden of the replicon is small. So they control their own copy number through a small RNA. So they have an interference, and then you have two RNAs, that one is for priming and the other for interference. And then this is a very low, so you can get high copy numbers with that. In fact, the coli 1 has been engineered, I mean, removing all the things and then just having this in a copy number control to be the PUC 19, PMB1 in origin, which has hundreds of copies. Okay, so but other copy numbers have proteins to do the copy numbers, so they have a genetic load. I mean, and these are transcription factors that usually they bind to the genomic DNA and non-specifically, and then this causes toxicity. So then more. So I think that it's and then. So it's complicated. So in general, there are papers, many papers about promoter strength and how it affects. So that would depend. But if you put a little and medium low promoter strength in a PUC 19, you don't clone that. So I mean, it's genetic. The metabolic load is so huge that it's unclonable. So. Okay, so I think that I don't see more questions, but maybe in the audience there are some more questions. Or I mean, or somebody wants to raise a hand, or I don't know.
Yeah, I think that were all the questions. I just want to say this is fascinating. I really enjoyed the learning capabilities in the games they played. Such a cool system.
Okay, thanks. So I think that I mean, this is something that later, I mean, is the simplest thing you can do. I mean, so that's I mean, of course, it's not that simple in the sense that if you want to model a complex behavior, then it's going to be not trivial. But I think that we use this additive system, and then so I mean it could be done in droplets, and then you don't need so you don't need even living cells. So then this could be done. Droplets also, I mean, have the nice facility. So you, I mean, you can create. You only need just having DNA replicating. Okay, so then and then more or less, and then translation. You have a translation system, but you can have that in droplets.
Yeah, that's true. You can. All right. Thank you so much. If there are no more questions, I just want to say thank you, Alfonso, again so much, and have a great rest of your afternoon.
Okay, thank you. Thank you to everybody for the questions and for the, I mean, whatever offline question. I'm open to offline questions, that you can send me an email or whatever. So, okay, so thank you very much.
Thank you. Thank you. Bye.
Bye-bye from Spain.